So let's get a little bit detail about the most common neurotransmitters. The first is acetylcholine. So acetylcholine is actually the first neurotransmitter identified and also the best understood. It is released by all motor neurons that stimulate skeletal muscle. We have actually talked about that when we talk about the somatic motor system. And also, it is used by most neurons in the autonomic nervous system, like all the presynaptic neurons are sympathetic or parasympathetic. And also, the postsynaptic neuron are parasympathetic, but not postganglionic neuron of the sympathetic. And also, acetylcholine is used by central nervous system neurons too. Acetylcholine is actually a quite simple structure. It's by one group called acetate, the other group called choline. So when bind together, it's acetylcholine. The choline and acetylcholine acted by an enzyme called the choline acetyltransferase. It's just one enzyme, one step. Once acetylcholine is released into the synaptic cleft, and there's an enzyme called acetylcholine esterase, or just called choline esterase, separate the acetylcholine back into choline and acetate. So as I just said, cholinergic neurons in the neuromuscular junction. A lot of drugs, especially we use like insecticide, is just targeting this region. Paralyzed muscle. So the animal, even the insects, they use muscle to do their job. So they cannot move and they die. A lot of biological weapon is also targeted on the neuromuscular junction. In recent years, the Alzheimer's disease patient, they seem to have fewer cholinergic neurons and acetylcholine in the central nervous system. So the central cholinergic neuron may be involved in this disease as well. When we talk about the autonomic nervous system, we already talked about preganglionic neuron and postganglionic neuron. Even they all use acetylcholine as the transmitter, but the receptor may be different. So there are two major types of cholinergic receptors. One is called the nicotinic receptor. This nicotinic receptor is like neuromuscular junction, like the most preganglionic neurons on the postganglionic neurons side. This nicotinic receptor itself is actually an ion channel. It's actually a sodium ion channel. Opening the channel, so cause sodium fluctuation, so depolarization and action potential. So there's a famous poison used by some South America tribes. They can put on their arrow. Shooting animals can paralyze animals. It's called the curare. And then there's a second type of cholinergic receptor called the muscarinic receptor. This is mostly a G protein coupled receptor, and they can be blocked by one famous drug called atropine. Now we get to the other largest group of neurotransmitters called the biogenic amines. They include dopamine, norepinephrine, and epinephrine, also serotonin and histamine. They all are derivatives of amino acids. Dopamine, norepinephrine, epinephrine, they are derivatives of tyrosine. And serotonin is the derivative of tryptophan, and histamine is the derivative of histidine. All these three amino acids are essential amino acids that are very important to our central nervous system. And these biogenic amines, they are widely distributed in the brain. They play many important roles in the central nervous system as well as the autonomic nervous system. So just give you a brief introduction how biogenic amines are formed. So they all started with tyrosine. The first step is 1-hydroxylation, so it's called the tyrosine hydroxylase rate-limiting enzyme. So then you will have dihydroxy. This is called the DOPA, so dihydroxyphenylalanine. And then dopa with the carboxylates. So you just remove this carboxyl group, then you become dopamine. Dopa is the precursor of dopamine. Then dopamine with another hydroxylation at the beta carbon region here, then this compound becomes norepinephrine. This is the major neurotransmitters of most postganglionic neuron of the sympathetic nervous system. And then, especially in the adrenal medulla region, they have another enzyme. And this enzyme actually is a methyltransferase that transfer a methyl group on the amino group here. So then you will have epinephrine. So norepinephrine and epinephrine. Difference is one methyl group. And this difference is one hydroxy group. And the difference of these two is one carboxyl group. So they are all very similar. Which neuron release or which chemical as their major neurotransmitter depends on the expression and the abundance of the enzymes. And actually, same thing can be said about serotonin and histidine. Tryptophan become hydroxytryptophan and become hydroxytryptamine. And that's serotonin, same as histidine. The neurotransmitters, functionally, they're just either excitatory or inhibitory. Or sometimes they can be both excitatory and inhibitory, depending on the receptor they are acting. Glutamate is one of the most important excitatory neurotransmitters. They always cause depolarization more than the others. And the same thing, inhibitory neurotransmitters also cause hyperpolarization than depolarization. And GABA, another amino acid neurotransmitter, and also opioids, they also cause depression. Then this hyperpolarization causes inhibition. And all the others, acetylcholine, norepinephrine, depend on the receptor they are acting. They are sometimes excitatory and sometimes can be inhibitory. And next, we're going to talk another category. They are not real neurotransmitters, but they also released by neurons. Sometimes they don't induce visible changes like APSP or IPSP. But their function is alter the effectiveness of the synapse. How they do that? They sometimes modify the postsynaptic cell's response to specific neurotransmitters. And sometimes they can change the presynaptic cells' synthesis, release, reuptake, or metabolism of the neurotransmitters. So that means in the presence of neuromodulator by itself, this doesn't do much. But with the presence of a neuromodulator, the more classical neurotransmission by the neurotransmitters, their effect can be enhanced or can be reduced. So they are modulator. So modulate the action of neurotransmitters. Now, we end the lecture today about the action potential, about the synapse, and about neurotransmitters. Thank you.